Okay, we're back to talk a little bit more about the cell. Our last PowerPoint video, we talked about the cell membrane or the envelope which holds the cell together. We talked about the cytosol or the fluid portion which is inside the cell, the liquid environment, and we also talked about the nucleus and nucleolus. But today what we want to talk about, we really want to talk about what are called the organelles. Okay, and the organelles, like we mentioned before in the last PowerPoint video, was these are these small little organs that are inside the cell that have a number of different specific functions that are really important for the cell to be able to carry on its normal daily activities. So let's talk about what those organelles are and what they really mean. You know, some of these organelles that we have we've already talked about the nucleus, so we won't spend any time on that, but the ribosomes. Ribosomes are placed for protein synthesis, okay? I'll just mention a little bit, and we'll talk a lot more about those each individually in depth, but ribosomes are placed for protein synthesis. That's where protein is made, in the ribosomes. If you have to remember something from this particular PowerPoint video, ribosomes make a place to make protein. Endoplasmic reticulum is sort of like a highway inside the cell. It's a series of channels and tubules inside the cell to let things get from one place to the other. Okay, and we'll talk about the difference between rough versus smooth endoplasmic reticulum in a little bit. Golgi complex is something else we'll talk about, and we'll spend some decent time on the Golgi complex. When the proteins are made by the ribosomes, they could be made for a lot of different things. They could be used for the cell, they could be used for parts of the cell membrane, they could be used in other organelles, or they could be exported and taken from inside the cell and taken to the outside. Well, to be able to do this, they have to be organized, they have to be packaged, and they have to be shipped. And that's the process of the Golgi complex. It does that, organizes, puts them together, decides where they're going to go, and ships them out. Lysosomes and peroxisomes are very similar. It's just that they are small little vacuoles. In fact, when we look up at the cells up here, see these little areas out here and maybe this out there, these little vacuoles, and what they do is they contain various enzymes. These enzymes are to digest materials that we want to get rid of, either old organelles that we want to replace with new organelles, or foreign debris that gets inside the cell like bacteria and stuff like that. They could be digested basically through various enzymes and materials that are inside these lysosomes and peroxisomes. Mitochondria. Mitochondria are really important because to make the cell work, it needs energy, and the mitochondria is a site of where energy is produced inside the cell, and that's what you need to remember. The cytoskeleton. The cells have a particular shape, and what really holds them together and also holds the organelles pretty much where they're supposed to be, sort of like the framework of the cell, is called the cytoskeleton. Our microtubules and tubules and things like that actually are strings that sort of like hold things, a fabric that knits the whole cell together. We also have what are called flagella and cilia. Cilia are little small hair-like structures on the outside of a cell for a specific purpose, and flagella are like whip-like tails. They're like hairs, but they're like whip-like tails that allow things to move like in spermatozoa. And then we'll mention just a little bit about the centrosome and centrioles, which are basically, we'll talk a little bit more about that in cell division, particularly the centrioles, when we talk about cell division in an upcoming PowerPoint video. So let's start first with ribosomes. I mentioned ribosomes is the site for protein synthesis, and the cells are loaded or jam-packed with these ribosomes. What we talked about before very briefly, and we'll be talking a lot about in an upcoming video, is that DNA has the code for how we make all these proteins. Okay, these are called genes. Along the chromosomes are various little sections called genes, and these genes have the specific code or the recipe to how to make protein. So they know how to make it, but where is it made? And the site of protein synthesis is in the ribosomes, okay? And these the proteins, like I mentioned a couple of minutes ago, the proteins can be used either by specific organelles. In other words, the organelles might need a specific protein to carry out their particular function, or sometimes the plasma membrane, because we know it's protein and lipid combinations, okay? And so therefore we might need some protein that's used in the cell membrane, or sometimes we just have protein that is going to be made inside the cell and shipped out of the cell to use somewhere else. Okay, we find that these ribosomes are basically two subunits that are made inside the nucleolus. Okay, and what they have is they have a high concentration of what we call ribosomal RNA. Ribosomal RNA, okay? And what this ribosomal RNA is all about, and we'll talk a lot more about it when we talk about how about DNA replication and the function of DNA, is that what they're responsible for reading the order of amino acids. Now, we mentioned this before. What's a protein? A protein is what's called a polypeptide, okay? And polypeptide means many peptides, and peptides are made up of amino acids. So basically, a protein is a string of amino acids that are then folded across and folded again and tucked inside and stuff like that. So we have a large molecule made from all these amino acids. Well, these amino acids are in a specific order in a specific location. Typical example, if I look at the normal hemoglobin, it's just an error in or misplacement of one amino acid because of an abnormal genetic code, okay, that results in not normal hemoglobin but sickle cell hemoglobin, which allows it to unfold. It's less stable, so it unfolds when there's a low oxygen tension in the air, okay. So what happens is the ribosomal RNA is responsible for reading the order of the amino acids from the code from DNA, so that the proper order of amino acids can be linked together to make a polypeptide or a protein. Okay, so we'll talk a lot more about that later on. So anyway, these two subunits of the ribosomes are made in the nucleolus, and they're assembled on the outside. Okay, and what happens is we'll see these. We'll actually see these. I'll show you some pictures of some ribosomes in a minute, some electron microscopic pictures of ribosomes. And what they do is some of them are attached to the outside of that nuclear membrane. Now, we talked about how the nucleus has a cell membrane or nuclear membrane around it, just like the cell membrane does, okay? So it has this nuclear membrane around it. So sometimes we'll see these ribosomes that will be attached around the surface. But then what happens is a lot of these ribosomes are attached to the inside of what's called the endoplasmic reticulum. Now, the endoplasmic reticulum is sort of like a highway. It's a series of channels and tubes throughout the cell, okay? And then what they do is you'll actually see these channels, and you'll see these spots on them, and the spots are actually ribosomes. That's called rough endoplasmic reticulum. We also have a number of ribosomes in the mitochondria, and they're in the mitochondria because the mitochondria, like we mentioned just a minute ago, is important for making energy. Cells need energy to be able to run, and therefore what happens is, to be able to do this, there are various proteins that are involved in producing that energy. So therefore we have some ribosomes that are also in the mitochondria. We'll talk a little about that later on when we get to the mitochondria. This is actually a picture of rough endoplasmic reticulum. Now, see all those little black dots? You know, if we draw those little dots, they're all over the place. All these little black dots all over the place. These are all ribosomes, and they're on these channels, or this highway, or these strands that are throughout the cytoplasm, and that's called the rough endoplasmic reticulum. This up here is the nucleus right here. You can see this double-layered nuclear membrane that's right there. So that's the nucleus, a double-layered nuclear membrane, and what happens is we see all these little dots all over the place, and these are all the areas. That's called rough endoplasmic reticulum. Rough endoplasmic reticulum. Okay, so that's the site of protein synthesis. Ribosomes, the site of protein synthesis, where protein is made inside the cell. Okay, and let's talk a little bit more about that endoplasmic reticulum while we're there. It's a series of folded membranes that start from the nuclear envelope, okay, or that double layer membrane of the nucleus, okay, and they channel throughout the cytoplasm, and basically they're for both storage as well as transport of substances around the cell. Okay, got to think about it. If I have this, if you can see a picture down here below, we see that the endoplasmic reticulum with all these little dots. Now those little dots are what? Of course, I just heard it, ribosomes, where the protein is made, which then could travel in multiple places, and they could be shipped throughout the cell. They're sort of flattened sacs or tubules that go all over the place. Okay, and you could actually see, if you look up here in the picture up here, these are all these the endoplasmic reticulum, all these folded membranes, and along them, it's hard to see in this picture, but there's small little gray colored dots, which are basically the ribosomes that would be attached to that. Now, there's two types of endoplasmic reticulum. One is called smooth endoplasmic reticulum. One would be smooth endoplasmic reticulum, and basically The smooth endoplasmic reticulum is endoplasmic reticulum without ribosomes. Rough endoplasmic reticulum is ribosomes with, or is the endoplasmic reticulum with ribosomes. Okay. So rough endoplasmic, this right here would be rough endoplasmic reticulum because we see all these ribosomes are attached there. Smooth endoplasmic reticulum does not have ribosomes. And because of ribosomes with the little dots, that makes it look rough. That's why they call it rough endoplasmic reticulum. Duh. Okay, got that figured out. Okay. Synthesis of fatty acids and steroids can be done in the smooth endoplasmic reticulum, as well as some detoxification processes. But a lot of the endoplasmic reticulum is basically going to be rough endoplasmic reticulum, okay? And so they're like a highway throughout the cell where they connect things to be able to transport materials and stuff like that throughout the cell through this endoplasmic reticulum. Now, if we look up in here, okay, these are some nice pictures, I think. What's this right here? This is going to be rough endoplasmic reticulum. Why? Because see all the little dots, all those little dots are the ribosomes. Dots of the ribosomes. Okay. If you look over on the picture on the right, on the upper right, hard to see, but if you look really close, there are little dots all over that. So this is still a lot of rough endoplasmic reticulum. If we look down in this area right here, most of this it's much more devoid of those little dots, and those dots would be the endoplasmic reticulum. So this would be your smooth endoplasmic reticulum right here. So anyway, that's what we have. So that's the difference between rough and smooth. Rough endoplasmic reticulum means it's these highways or these tubules that are coated or dotted with all the ribosomes, where the smooth endoplasmic reticulum is basically for other things, for lipids, for detoxification, stuff like that. And it's not the site of protein synthesis because there are no ribosomes. Okay. And again, the parts of the ribosomes are made in the area of the nucleolus, and they're extruded out. They'll be along the surface of the nuclear membrane and then along the rough endoplasmic reticulum. Okay. Now we get to the Golgi complex. Again, we talked about this Golgi complex, and what it is, it's for once the protein's made, okay, it just can't sit there. You have to, if you're making it, you know, it's sort of like if you make a cake, you don't let that cake sit there on your counter for weeks and look at, oh, that's a nice looking cake. I bet you it tastes really good. You know, you got to eat the thing, okay? So you have to figure out what you're going to do with the cake once you make it. You also have to figure out what you're going to do with the protein in the cell once you make it. So basically what the Golgi complex is for packaging, processing, sorting, and delivering the proteins and lipids throughout the body, okay, or excuse me, throughout the cell, okay? So it's actually the first step in trying to figure out where these materials are going, well, first of all, what they are, how they might be assembled with other things, as well as where they're going to go. Okay, so it's the first step in that transport of substance in the pathway of the cell. Okay, they're basically, again, flattened membranes with bulging ends, and most cells have multiple Golgi complexes within them. There are multiple areas with the Golgi complex. They have what's called an entry and an exit phase. Okay, guess what those mean? Well, the proteins and the materials go in at the entry phase, and they come out packaged and processed at the exit phase. You know, the entry phase is sort of concave. It faces the rough endoplasmic reticulum. Why? Because that's where it's getting all these proteins. The exit phase usually faces the plasma membrane. Why? Because some of that protein and the materials that are actually packaged and sent out through the Golgi complex are going to be leaving the cell. They're going to be taken, they merge with the cell membrane, and then the cell membrane sort of like spits them out. Okay, so the major function of the Golgi complex is the modifying, sorting, packaging of proteins for secretion or for use somewhere in the cells. Okay, it's also involved in the transport of lipids around the cell, the creation of lysosomes, which are basically various enzymes. Okay, or the lysosomes are actually like bubbles within the cell that have the enzymes inside of them, the digestive enzymes. Okay, and what happens is when I show you some pictures of what the Golgi complex looks like, they look like little folded sacks. You know, look like you know, something that's folded over and folded over and folded over and folded over and folded, like an accordion, you know, type of paper. And that's what those Golgi complex. So basically, the Golgi complex is like the post office. You drop off a letter, you drop off a package, the guy says, is there anything liquid, perishable, flammable, explosive, and stuff like that? You say no. He pressed a little button on the little machine, says no, that you accept that, and then he takes it, and then he puts a little sticker on there where it's going to go, and he takes it and heaves it in a box and breaks and stuff like that. No, it doesn't really do it. They actually do a reasonably good job. Okay, but anyway, but then they send it to the back. You never see it again. Eventually, what happens is they say where it's going to go. They put it in the right pile. The right pile gets done. Then they stick it on the right truck to go to the right place to be delivered to wherever it has to go, and it's shipped out. It goes out the back door. Go into the front door. So here's my entry face right here. The exit face is where the truck's leaving in the back. Okay, and basically that's a Golgi complex. Again, what we just sort of like just to fill you into this a little bit more. You know, proteins are synthesized by the ribosomes on the rough endoplasmic reticulum. We better know that by now. Okay, and what happens is once they're made on the rough endoplasmic reticulum, part of the membrane of the rough endoplasmic reticulum folds around them, around these proteins. Okay, and sort of like encloses it in like a little package. Okay. So what happens is that package then leaves and goes to what's called the entry face. This would be the entry face right there of the Golgi complex, okay? And what happens is these little bubbles with the proteins in there, they get to the entry face and they merge. They stick to the entry face, the membrane, and what happens is it merges. And then what it does is deposits what's inside there, okay, inside that entry face. It deposits it into the channels of the Golgi complex, okay? So they fuse the vessels of the entry face, release the proteins into the Golgi complex. So the Golgi complex then work down this whole tubule into that through those cisternae, okay? And what happens is, depends upon what they're being packaged with, they may be manufactured with other materials to make into things like glycoproteins, glycolipids, lipoproteins, and stuff like that. So these materials, whether it be a lipid or whether it be a protein, are transformed. There's things added to them. There's other things pushed together, more like an assembly line, maybe in like an automobile plant making cars. Okay, and that's what this does. And then finally, what happens is they finally get to the exit face, and the exit face would be out here. And what happens? They're actually put again back in little packages, and they're extruded to the outside wherever they need to go. Okay, so they're going to be shipped at that point, and that's what the Golgi complex does. It organizes, it transforms, modifies, combines it with other things, packages it, and ships it. So it has a lot to do in there. So here's some boxes being packaged and shipped to where they need to go. Now this is a Golgi complex. So we see, if you look this area right here, this is a Golgi complex right here. So this would be the entry face. This is the exit face. Okay, coming out this way. So what happens is this is the mitochondria. Actually, no, I'm wrong. This is the entry face, and this is the exit face. So what happens, because this is the concave side. So this is a nuclear membrane sitting right here. Proteins are made. Here's all these little, what do you think these little black dots are? Those are ribosomes. They're sideways in the endoplasmic reticulum. What happens? Here's vesicles that are entering the area of the phase and into there, gets inside there, and it's processed, and it's then eventually extruded out to the back end. What are these over here? These are called mitochondria, and the mitochondria then might need those proteins. Okay, that's what we see with that. Again, here's a Golgi apparatus. Got things will enter here into the entry face. Exit face would be out this way, going to the outside. Here's lysosomes. Again, they're making some of the stuff for lysosomes and helping to make the lysosomes, which have enzymes in them. But this is a Golgi complex, okay? And that's just with the Golgi complex. Just again, sort of review. Again, what happens is this shows what happens is here's the rough endoplasmic reticulum. There's the ribosome. These are all little ribosomes in there, and they make the proteins. So you see the proteins right there, a little squiggly little thing. Those are those polypeptides chain that's folded around on itself of various amino acids. It's put into a vesicle. There's a vesicle. Vesicle gets to the entry face. So here's my entry face. Right here, gets the entry and fuses with it. So once it gets there, it fuses, and then now all of a sudden it gets inside that cisternae, which is basically this large trapped area right there. Inside there, it's changed, it's modified, does do other things inside there. Finally, at the exit face, it's put into another vesicle and it's gone out wherever it needs to go. You know, this transport vesicle goes to lysosome in here. This one maybe goes to the cell membrane for either incorporation or for eliminating it from that, or this goes to other places like mitochondria and stuff like that. So basically that's what we have, okay? So some are processed, leave the entry phase for secretory vesicles, the plasma membrane for exocytosis. That'd be in here. Other proteins leave the exit phase in the membrane for plasma membrane, and other ones leave to go other destinations inside the cell. So that's what the Golgi complex does. Mitochondria. Mitochondria, like I mentioned before, is the powerhouse of the cell. It's what needs, it makes the energy that's needed for the cell to be able to run its daily function, okay? It's the site of making what we call ATP. ATP stands for adenosine triphosphate. And what happens is this is a molecule has 3 phosphates that are attached to it, and they're called what are called high energy bonds, okay? And what happens is ATP then, when it needs to release energy, one of those phosphate bonds is released, which then causes a release of energy to the outside. And then ATP, okay, ATP then becomes ADP, or adenosine diphosphate, so tri-3,3 di-2. So and then as a result, energy is released. Energy released. And that's how that works. Okay, so there are many mitochondria per the cell. Why? Because we need a lot of energy, okay? And so the cell, and some cells have a lot more than other cells. Let's take an example. Let me just take muscle, skeletal muscle. Skeletal muscle is constantly working. Even when we're asleep, the muscles have a little bit of tone, which means they're always mildly contracted, just a little bit. So as a result, because of that activity, they always need energy. And so therefore, there's many mitochondria. Liver is, I'm tell you, you know what? If I ask somebody what your favorite organ is, everybody's going to say the brain or the heart or something like that, you know. Or if you like to eat, maybe the stomach might be important to you. Nobody mentions the liver, okay? Liver is the unsung hero of the body. It does so many things. It's metabolically detoxifying things. It's metabolically activating things. It's making clotting factors. It's making vitamins. It's doing so many different things. And so the liver, because it has so many things going on, the liver is the absolute 100% multitasker. Things are going on all the time in the liver, okay, and different things. So guess what? It needs a lot of energy. So therefore, we have a lot of mitochondria in liver cells, okay? So the highest concentration of mitochondria are in areas where we need a lot of energy production. If we need a lot of energy production, that's where the mitochondria seem to be in the highest concentration at, okay? Oh, there we go. Go mitochondria. You betcha, okay? Yeah, making that energy. And what happens is there's an outer mitochondrial membrane, okay, around the outside. And then inside, what we have is these little folds, okay? These folds inside the mitochondria, inside the mitochondria are called cristae, okay? And this is something that we're going to see as a recurrent theme in anatomy and physiology for the rest of the semester. What happens is, the reason why there's all these folded membranes on the inside of the mitochondria is, number one, it increases surface area. And by increasing the surface area, it allows more areas in which energy can be produced. Okay, we're going to see that in this inside the intestinal wall. Okay, and the intestinal wall has multiple little fingers called villi, and because you can't see them, they're microscopic, but these little villi increase the surface area. Instead of being a flat surface, now it's sort of like, you know, all these little fingers. Okay, and therefore it increases surface area. By having all these little folds in the mitochondria, these cristae, what happens? It provides an extensive and increased surface area for these chemical reactions to occur with cellular respiration. Cellular respiration means producing energy within the mitochondria. Okay, so therefore it could produce, you know, significant amount of ATP because it's increased that surface area for more areas for these chemical and metabolic processes to occur. Okay. Besides the cristae, there's the matrix, which is the large central fluid-filled area surrounded by an inner membrane. So you can see the inner membrane, you know, if we're looking here, here's the inner membrane right there. Here's the outer membrane, you know, Here's the matrix in this area, and here are all those little cristae that are all over the place. Okay, there are some ribosomes in the matrix for protein synthesis as well. So sometimes they need to make their own protein. There again, what's that protein used for? For certain processes that might be chemical processes that might be needed to be able to let the mitochondria make energy. Okay, so that's what the mitochondria do. This is just a couple beautiful pictures of mitochondria. And remember we talked about this being the Golgi, but here's a mitochondria right here. You see those little folds? Those are all the cristae. Here's a little mitochondria with all the folds. This is a great, I mean, this is one you might want to just, you know, if you're looking for another tattoo or something like that, just to get that tattooed. And this is the mitochondria with all those little folds, and there are all those little cristae. And these are, so what do you think these little black dots are? Of course, you know, those are little mitochondrial ribosomes. Okay, so basically that's what we see. This is mitochondria. Okay, nice picture of the mitochondria. Really nice picture. Okay, lysosomes. Now the cell needs to get rid of things. Okay, it's not the cell's not really much of a hoarder, you know. So what it does is that things that get inside the cell get engulfed for one reason, like bacteria and materials like that. Also parts of the cell, organelles, that are worn out, that are no longer working, they will take those organelles and just dissolve them. How do they dissolve them? Because inside these lysosomes are all these digestive enzymes. These enzymes will actually sort of digest things that it needs to digest. The cell also will sometimes just auto-destruct or self-destruct after a certain point of time when it's reached a certain limit. Cells are programmed that they get to a certain age and they will automatically destruct themselves simply because they're past their use. They can't, they're taking up space and not doing their job. Okay, so basically these lysosomes contain digestive enzymes. There are more than 60 different types of digestive enzymes in these lysosomes, okay, as well as hydrolytic enzymes, okay. And again, this is just to break things down, okay. Also, the environment of these enzymes is acidic, okay, and the acid just makes things break down a lot more. I don't know if you've known anybody in your family or a relative or something like that, or you know, next door neighbor that always wants somebody to take his dog out or something like that, but they may say, you know, I'm having an episode of gout, you know. And what happens in gout, it's interesting. Gout is because of a high concentration of what's called uric acid. And uric acid is a crystal. And these crystals look like little spears, okay? It's just a little spike. It's like a little needle, okay? And what happens is when and this happens usually in the big toe joint, but can occur in other joints. And what happens is the uric acid will precipitate, will start to accumulate in some of these joints. And what happens is the body says, that crystal shouldn't be there. It should not be there. And what happens is it sends white blood cells to take them. So the white blood cells go down. So let me just draw a white blood cell. Here's a white blood cell somewhere here. Here's a white blood cell coming down here. And white blood cells moving closer and closer. And eventually this spear this crystal like a spear, guess what it does? It goes right through the white cell. It punctures through the membrane as the white cell's trying to eat it, okay? What happens is the crystal goes on through. What it does is as a crystal, like a spear, goes through that white cell, guess what it punctures? Whoops, it punctures lysosomes. It also destroyed the cell membrane here, destroyed the cell membrane here, and guess what happens? The cell's going to die, and what's going to do, it's going to release all these lysosomes to the outside, which then causes that intense inflammation that we see in people with gout. I mean, it's like I've seen hundreds of people with gout over the course of 25, 30 years, and these people, it looks like they're like on fire. You actually put your hand really close to like their foot. You can actually feel the heat radiating off, and that's because these lysosomes, they're supposed to do a good thing. What they do is, because there's so many of these crystals, all these white cells start to eat them up. It spears and destroys the cell. Lysosomes are released to the outside that causes an intense inflammatory response. Okay, so anyway, the lysosomes are digestive enzymes. They're there to digest things that either wants to get rid of, either foreign invaders that are getting into the cell, or itself, you know, if it needs to when the cell reaches its age limit. Okay, ooh, there's the lysosome. Oh, it's sort of like dissolving that little thing. Oh, yeah, let me do this so you can see it better. I want you to make sure you see this, okay? Watch, here it comes. So there's a bad guy right there, and the lysosome. Oh, we got him, okay? And there he goes. Now the lysosome's taken care of, okay? Peroxisomes. Peroxisomes are really similar to lysosomes, but what they do is they contain what are called oxidases, okay, oxidases. And oxidases, if you see something that ends in ASE, it usually means that that's an enzyme. An enzyme, I mean, not all words that end in ASE are enzymes, but if you see ASE, it usually means it's an enzyme, okay? And these oxidases oxidize various organic substances. So they're very similar. They also have enzymes, but what happens is because they're they're called peroxisomes, okay? And again, things like amino acids, fatty acids are oxidized. That's how they get rid of those. Also, things like alcohol and stuff like that, other toxic substances are oxidized. So the peroxisomes will take care of them, okay? Here's one of the problems with peroxisomes, okay, is the byproduct is peroxide. Well, say peroxide, I put that on my wounds, I get a cut, I get a boo-boo, and I put a little peroxide in it, and it bubbles up and looks nice and cute and stuff like that. Well, actually, the peroxide inside the body is toxic, okay, and it's actually may cause cellular destruction, okay, and so you have to watch some of these, because what happens is you don't want all this peroxide, because the peroxide will actually destroy a cell as well. So the byproduct of this is peroxide. Here's what the cells do, okay, to make sure that the hydrogen peroxide that's produced as a byproduct of the peroxisomes doesn't cause a problem. There's also an enzyme there called catalase. Catalase decomposes the H2O2, which is peroxide, okay? Now, let me ask you a question. If you ever put peroxide on a wound, okay, what did you notice after you put the peroxide on the wound? Just tell me. Go ahead. I'm waiting. I'm listening. Come on. Oh, you saw it bubble, okay? And why it bubble? Because when you get a wound, it releases an enzyme called catalase, and that catalase takes and decomposes the peroxide, and H2O2 gets converted to H2O plus oxygen, and eventually with a couple of them it becomes O2. So what's this? Water. What's that? Air. What's that? Bubble. So when you take that peroxide, what it basically does is the tissue catalase from a wound will actually decompose the H2O2 into H2O and water, or H2O, water, and oxygen, and that's why you see the bubbles. So I never used peroxide as a doc. I never used it a whole lot in regards to an antiseptic, but what I called it more as a debreeder. So if a wound was dirty, you could put some peroxide in it, sort of bubbles all that dirt and grime and stuff like that out of it, because the catalase in the tissues in a wound will actually decompose the peroxide, because another peroxide is what? Toxic. Okay, so it's probably a good thing. And these are just pictures of peroxisomes and lysosomes, and basically you don't see a whole lot there. Basically, here's a mitochondria up there. There's a mitochondria right there. So forget that. Let's draw that. We'll get rid of that. And there's a rough end of forget the rough. There's a lysosome. That's what we want to know. So this area right here, the lysosome. Yep, that's what we want to look at. Here's a lysosome. Here's a peroxisome. Here's another peroxisome. You know, and there's a lysosome up in here, and basically they're just vats, they're just bubbles that have these digestive enzymes that can be used to destroy things that it wants to get rid of, either parts of itself or materials that's engulfed. Okay, let's talk a little about the cytoskeleton. What happens is the cells have structure, okay? Cells are able to change shape a little bit, but you need sort of things held in a certain place, and you need a certain structure, okay? And the structure that we find in cells is what we call a cytoskeleton. And that cytoskeleton is basically what provides a structural framework for the shape and the organization of cellular content. It gives the cell a particular shape. When we talk a little about tissues, when we talk about the microscope, when we look at things if you look at things in the microscope, which we don't have a microscope, so we'll be looking on the screen. When we look at things under the microscope, what's going to happen is you will see things are held. The cell will have things are held in certain locations. Plus, the cell has a certain shape. okay? And this certain shape is a lot because of what's called the cytoskeleton, okay? Also what happens is inside a cell, there may be other types of filaments, part of the cytoskeleton that goes from side to side, top to bottom, front to back, whatever the case, and actually allows the organelles to be sort of like hang from them. And as a result, it keeps those organelles in their place. Okay, so it sort of helps organize things a little bit by the cytoskeleton. Now, what do we have in the cytoskeleton? We have what are called microfilaments. Okay, we have intermediate filaments and what are called microtubules. Okay, and we'll talk a little bit about them because you don't see a whole lot of them when you look at the cell. These microtubules are basically hollow tubules. Makes sense. Here's a microtubule here. They grow out from the centrosome. We'll talk a little bit more about this. Okay, what happens is when this is going to be more important. This centrosome. We're going to talk about this when we talk about cell division a little bit more. Okay, centrioles and centrosomes. So think about that now. But what happens is it allows what the centrosomes and centrioles do is they create these tubules that come out from a center area and allows in cell division the chromosomes to line up there so they can pair up, so they can divide. Okay, we'll talk more about that later on. They're much more important in cell division, these microtubules. Okay, but they also help to determine cell shape, the function and movement of the organelles. They participate in, again, the movement of chromosomes, like I mentioned before, in the cell division and stuff like that. They're also part of what we see as called flagella and cilia, which we'll show you in just a few minutes. We have intermediate microfilaments, and these are very thin, small filaments. Okay, and basically they're mostly for stabilization for the position of the organelles, basically like a lattice work on which things, and they're basically a protein themselves. And what happens is they could also generate movement and provide some mechanical support to things. They're not, you know, of all the organelles in the cell, they're probably one of the least attractive. Not attractive like they don't have beauty, because they do have beauty, but it's sort of like the least interesting, sort of. But even though they do have a function in regards to structure as well as organizing things inside the cell. These are microfilaments and microtubules, and that's what it looks like inside there. And basically now the organelles would be hanging from this here, and there's a mitochondria sitting right there, and whatever the case may be. So they give that cell the shape and structure, as well as they allow certain organelles to have stuff. And this is what we talk about, the centrioles and stuff like this. This is cell division. We start to see cell division right here, and what will happen is we have these centrioles, and what happens is from the centrioles, which is the center, they have these microtubules that come out. You see all these little microtubules, and this is allowing the chromosomes to be able to line up. And what happens is this is earlier on, so these centrioles, there'll be a little area right here, and whoops, here and here, and there's these little tubules that come across this way. Okay, and these are all little chromosomes that are starting to line up. Eventually they'll split. Now this cell is about ready to divide into two. This one's getting ready, and this one's getting ready to divide into two. So now all of a sudden these chromosomes will move along these tubules, and they finally will congregate into the area which will eventually become the nucleus on the two opposing cells. Okay. The last thing I want to talk about today, well, I got two things, cilia and flagella. Okay. And what happens, these are small little projecting things that come out from the wall of something, or a flagella would be a whip-like tail. Cilia are these little short projections. I'll talk about those, and so I'll show you some pictures of what they're like. And they're basically a specialized arrangement of contractile microtubules. Okay, now this is where they become interesting, and they project out of the cell surface, and they're for one of two reasons largely. Number one, to increase surface area, such as we see with cilia, or number two, to produce motion, what we see in flagella. Okay, cilia, like I talked about before, are little small projections that hang out from the side of a cell. What they do is they have they beat. They have like a little beating action to them. Let me see, there you go, there they beat, they have a little rhythm to it, and what happens, it allows things to move along the surface of a cell. So they're on the outside of a cell, and they allow things to move along the surface of a cell. Like I mentioned, the intestine has folds which are called villi, and on the outside of the villi are small little things called microvilli. Okay, but that's increases surface area. But where we see cilia largely is in the respiratory tree. Larger airways are lined with these small little projections, these little oar-like beating projections come out from many of the cells that line the tubules, the bronchi, and some of the larger, bronchioles. Bronchioles are starting to get muscular, so we're starting to lose some of that. But mostly the bronchi, okay, with these little cilia, and they trap material that we breathe in. So when we breathe things in, okay, when we take a deep, okay, here's what I want you to do, okay, ready? When I say go, take a deep breath, go. Okay, take a deep breath. Okay, humor me, try it. I can't tell if you're doing it or not because my screen doesn't get sucked in or anything like that. But what happens, if you took a deep breath, what happens is, I hate to tell you this now, you've breathed in all kinds of garbage, particulate matter, I don't care how clean you are, dust mites, little bugs, all kinds of stuff that you're breathing in. So what happens as it's going down through the airways, it gets trapped in the cilia. The cilia will collect them, okay? They sort of like hang out there and they sort of, grab them a little bit, and so they collect all this garbage that you're breathing in. And what they do is they beat. So they beat. What they do is they transfer this stuff. They like body surf these little particles. They body surf them, body surf them, and finally they get up to right where the trachea branches off into the bronchi, right about this area in the chest. Goes to the right bronchus, goes to the left bronchus. And what happens is when they branch off, right at that area, it's called the carina. It's like an upside down V. We'll talk about that when we talk about the respiratory system. There are special cells in there. And these special cells are actually the origin of a cough reflex. So what happens is when we breathe in all these particles, that what happens is they get trapped on these cilia. And the cilia beat, when they beat, they move them towards this area of the carina where the trachea bifurcates, divides into the bronchi, and at that point when they get there, they irritate these cells. These cells say, hey, let's get rid of them, let's cough them up. You cough that puppy up. Okay, so anyway, that's the cilia. We also have flagella, and flagella are, instead of like small little projections, they're more whip-like tails, and they're basically for cell propulsion. Okay, spermatozoa an ovum for fertilization only goes a short distance down the fallopian tube. It just sort of rolls like a ball. And you know what? Oh, this is a good point. You know what actually rolls the ovum down the fallopian tubes? The ovaries are produced, the ovum are produced in the ovaries, and they're sucked up into the air of the fallopian tubes. And what happens is they start to go down the fallopian tubes. And you know what makes them go down the fallopian tubes? If you said flagella, you're wrong. It's cilia. There's cilia in the inside of the fallopian tubes that actually start to move the ovum and sort of transport it down the fallopian tube. However, they go a short distance. Spermatozoa have to go a long distance. How do they go? Because the spermatozoa have a long whip-like tail at the end of the spermatozoa, which is called a flagella, and it whips back and forth to allow that little puppy to swim through the water like a little tadpole. Okay, so that's what the flagella is really as compared to the cilia. And if we look up at the top left, that's basically, we see with the cilia. That's the surface of a cell. That's a electron microscopy picture, and all these little things are all these little hair-like projections, which are called cilia. And you can see it looks like a artificial turf that's there, but they're microscopic, obviously. And what happens is all these little particles will get trapped inside there, and these little cilia will beat to be able to move those particles where they want them to go. Okay, so that's cilia. On the bottom down here, these down here would be the flagella. It's a flagella here. Flagella. This is a spermatozoa. That's the head of the spermatozoa. That's the area that has the chromosomal material. And this is basically a whip-like tail that allows it to swim. Okay. So up here is the cilia. On the upper left, down lower right, basically spermatozoa. There's the head. There's the flagella, or the flagella. Here's the head. There's the flagella. Comes out this way. Okay. And basically that's what we see there. You could call it cilia. I prefer to think of it as designer stubble. Okay, so maybe that's again, a little, somebody dyed it green a little bit. It's a designer stubble. He told you what? He's pulling your flagellum, Nancy. So anyway, that's the flagellum that's coming out there, that long tail. Okay. Should have said Robert or something like that. Keep your flagellus to yourself, children. So obviously that's a school for some types of protozoans with flagellas. One last thing that I want to mention here before I let you go and bore you any more than we have today are what are called inclusions. Okay, and sometimes inside the cell we have things that are going to be needed. It's like a storage area. It's a storeroom, and these inclusions are basically storage areas, storerooms for things such as glycogen. Glycogen could be used for energy production. Glycogen is actually a string. It's a carbohydrate that's a long string made from glucose, made into long like string. The body can't really store glucose, but what it does, it converts that glucose into something called glycogen, and glycogen can be stored in muscle as well as the liver. So as a result, what happens is, if we're looking inside muscle cells, there might be inclusions with glycogen. Also melanin. Melanin's a pigment. Okay, when we look at the pigmentation of the skin and things like that, at one of our lower layers of the epidermis, you look inside, there's these little dark granules. These little dark granules are melanin pigments, which will give pigmentation to the skin. Also triglycerides, which could also be used for energy and stuff like that. So basically, substances that are produced by the cells, but they're really not bound by a membrane. They're just clumps of them, and they're called inclusions. Okay, and so that's just a couple examples of what we see with inclusions. Okay, and they're all over the place. So what have we talked about today? We've talked about, the organelles. Okay, you have to get out of this with at least a couple of the major points that we're talking about. You have to talk about the fact that the ribosomes are the place where we make proteins. Okay, and again, we're going to be talking a lot about ribosomes when we talk about DNA and RNA function, because again, what does DNA mean? It has genes on it. What are those genes for? The genes are codes for making proteins. Okay, various proteins, hundreds of thousands of proteins. Okay, are in each of those little recipes in what we call a gene. Okay, so ribosomes for making protein. Endoplasmic reticulum, for a way for things to get around the cell, to move around the cell, through that rough endoplasmic reticulum. It's a place for the ribosomes that are sticking to the endoplasmic reticulum to make protein. Sort of like going through a carnival, and you look on one side and you got somebody making elephant ears, you got somebody on the other side French fries, another side with burgers and stuff like that. So it's basically, you're walking down that aisle, and that aisle is the endoplasmic reticulum, and all those little food stands are like the ribosomes making all that protein for you to eat. Okay, so that's what the endoplasmic reticulum is. It's a way to get around rough endoplasmic reticulum is endoplasmic reticulum with ribosomes. Smooth endoplasmic reticulum is that when you get to the end of the carnival and there's no more food stands and it's just a walkway. That's the smooth endoplasmic reticulum. Golgi complex. We know that once these proteins are made, they have to be processed. They have to be analyzed. They have to be processed. Some of them have to be remanufactured in certain ways, or adding more things to them. And then what happens is, once they're finally done, they have to be shipped. And that's what the Golgi complex does. It processes, manages, organizes, and finally ships the materials to where it needs to go throughout the cell. Okay, mitochondria. Mitochondria are there to make energy. That's their big purpose, make energy. Why? Because the cells need energy to be able to survive. So we need those mitochondria. Cytoskeletal structures basically are to give the cell some shape, or to allow something to hang the organelles from. Okay, cilia and flagella. Cilia and flagella are basically those little, either small little fringy things that will trap material, such as in the cilia, or longer, like whip-like tails, will allow things to move around for propulsion. Lysosomes and peroxisomes. We know we have to get rid of the waste. Okay, and to get rid of the waste, how do we do it? We digest it. So what happens, these lysosomes and peroxisomes have various enzymes that will convert it to stuff. And what happens is, then the waste products then are put together into what are called vacuoles, or excretory vacuoles. And what they do is, they actually will start to float to the area of the cell membrane. They merge with the cell membrane, and the cell membrane just spits them out, okay, to the outside. And then forget it. Once they're gone, it's like, throwing the garbage out. You don't care which landfill it goes to, it's just going. Okay, so basically that's lysosomes and peroxisomes. And inclusions are just clumps of material that are in the cell that aren't really bound up to anything, but things that could be used, like a storage area, where if I need something, it's right there, just grab it, boom, it's ready to go. Okay, so hopefully you got a little bit of what we talked about today in regards to some of these organelles. And if we combine that again with what we talked about last time in regards to cell membrane, nucleus, nucleolus, and cytosol, that gives you a lot. Now, we still have some things to talk about in regards to the cell function, and the big thing we have to talk about next, it will be talking about cell membrane. And what does a cell membrane really do, and how does it work? Because the cell membrane, all these things that we make, such as in the Golgi complex and stuff like that, and materials that we have to get rid of. How do we get rid of things to the cell membrane? How do I bring things in to the cell from the outside? Because obviously you might have to, Amazon doesn't ship to the cell, so I have to get there some way. How do I get it from the outside, the extracellular, outside cellular, outside the cell, to the intracellular, inside the cell? How does it go? And that's what the cell membrane is all responsible for, and we'll be talking about that next. Okay, so next PowerPoint video, cell membrane dynamics and cell membrane function. And we'll just something, make sure you bring some Red Bull or something like that, because my gosh, it's you're gonna have to keep your mind going. Okay, whatever's going to stimulate your mind, you have to think about it. So I'll see you next time. Okay, be good.